stem cell, in this case an embryonic stem cell. We know they're pluripotent and that you can culture them in vitro, and if you figure out the conditions just right, you can differentiate your embryonic stem cell into any cell type. And so, for example, here they're showing blood cells, neural cells, and muscle cells. And the idea is that you could take these embryonic stem cell-derived muscle cells and transplant them into a patient that needs to regenerate muscle, maybe because of disease or because they're old or because their muscles just decide to fail, and that these cells will integrate with the damaged tissue, replace all the damaged tissue, and regenerate fresh new muscle cells. So that's the goal. And Jill brought this up during discussion. So when we talked about embryonic stem cells and to test whether they're pluripotent or not, we know that when you put them in a blastocyst and then take the blastocyst and put it into a surrogate mouse, you'll get normal embryonic development, and all those embryonic stem cells will integrate properly into the germ layers of that embryo. But if you just take embryonic stem cells in culture, undifferentiated, and inject them into a mouse in the skin, then you get a teratoma, or this is a teratocarcinoma, which is also teratoma. Okay, so as she said, that's a problem. You don't, if you were going to do a stem cell derived therapy, you don't want to give the person a tumor while you are trying to regenerate their damaged cells. So one of the biggest issues with embryonic stem cell derived therapies is that you need to figure out that whether the cells are well differentiated before you transplant them in. And so there's a lot of work going on in many different labs to figure out how to differentiate an embryonic stem cell into this tissue that you want before you transplant. Okay, but if you can figure that out, then there's a lot of potential therapies that can be designed. So you could take an embryonic stem cell and you could turn it into skin or brain cells, and this could help patients that maybe have Alzheimer's, or burn patients if you want to make artificial skin. You can take make endodermal cells, which would produce, say, for example, pancreatic cells that could be important for diabetes patients. You could replace lung cells, which would be important for patients with cystic fibrosis. And then you can also make mesodermal cells and try to differentiate, say, heart type cells, heart muscle, regular skeletal muscle, blood cells, etc. So there's a lot of potential for the use of pluripotent stem cells for medicine and tissue repair. And the idea is that eventually you should get some kind of personalized treatment. So the whole question of whether or not to use an embryonic stem cell or an adult cell and reprogram it can also feed into this, into a medical application. Okay, so obviously it's not that easy. Okay, so this is an example of how to turn an embryonic stem cell into mesoderm or endoderm. And so you can see that all of this information is basically, we know all this information based on the regular developmental biology studies. So we know that embryonic stem cells in a mouse will incorporate into an epiblast. We've talked about how BMP4 and Wnt and activin are important for production of ectoderm into skin or neural cells. If there are other signaling factors that can produce more mesodermal type cells. And we know that in the different germ layers, there's different signaling pathways that are activated and inhibited. And in the end, we can use this information to produce very differentiated cell types. So what we need to do is now figure out how this works in vivo and then apply it to an in vitro culture. So we can use developmental biology to inform how to manipulate embryonic stem cells in vitro. So this is why we study kind of the production of one cell type versus another cell type. So this is not without controversy. Okay, so there is the question of, well, if you use embryonic stem cells or should you use adult stem cells? And so this is a cartoon from the newspaper that shows an older angel. It says, I died waiting for embryonic stem cell research to find a cure. What about you? And then here's a younger angel that says, well, I was the embryo. So you have both sides, right? You should see this and you should feel conflicted. So in response to this controversy, we are looking now at induced adult pluripotent stem cells. And that's the paper that we read today. This is a review article about the science of it. Again, it's just a review of the different terms. Okay, so there's totipotent, pluripotent, multipotent, unipotent, reprogramming, transdifferentiation, and plasticity. And so you should be familiar with most of these terms. So people have been talking about trying to make adult stem cells into more or less differentiated cell types for a long time. We've already talked about the strategy of nuclear transfer. We've talked about cell fusion. And today in discussion we talked about infection with virus. So there's been a lot of ways to reprogram somatic cells. But there's a lot of challenges to using them for medicine.